and Dr. Vijay Singh sir. The session of yesterday was very insightful, inspiring, and faithful for us. I believe we can see and feel the very same today. On behalf of the MIT Department of Pharmaceutical Technology, I, Aishi Khurana, Assistant Professor, welcome our first speaker of the day. Due to some issues, Dr. Dhiren Kaushik was the first speaker of the day, but due to some issues, he would not be able to join us today. So, Dr. Shivkant Sharma sir will take lead today in place of him. I want to introduce about Dr. Shivkant Sharma sir. Dr. Shivkant Sharma is presently working as an assistant professor in Department of Pharmaceutical Sciences, Gurugram University. Dr. Sharma holds position of Central Library Convener, NCC Coordinator in charge, Department of Physics in charge, Department of Social Work, member, PG Board of Studies of Pharmacy, Neuroscience and Public Health Department of Gurugram University, Gurugram. He earned his doctorate and master degree in the field of pharmacology from Kurukshetra University, Kurukshetra. He has been awarded with University Research Scholarship for pursuing his PhD and secured first position in pharmacy in university. Dr. Sharma has 9 years of teaching and research experience. 4 pharmacy and 3 masters of public health students are doing their dissertation work under his supervision. His research area includes molecular pharmacology, clinical pharmacology, toxicology, developmental toxicology of pesticides. He has 10 research publications in national and international journals of repute. He has attended more than 35 national and international conferences, workshops, seminars, and successfully presented 15 papers as oral and poster presentation. Dr. Sharma is a life member of various national bodies of pharmacy, including APTI, IPGA, IPS, etc. I welcome sir on behalf of the MIET. Please, sir. Please. I would like to welcome sir.
He's not online. I think some network connection. He has just left the meeting. I think he must be rejoining. Oh, he's back now. He's back.
Okay. 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 Sir,
yes, ma'am.
Am I audible, sir?
Yes, right now you are audible due to.
Oh, okay. Sir, I would like to welcome, yes, I would like to welcome our first speaker of the day. Please, sir.
Dr. Shiv, you can start your session.
Can I start, sir?
Yes, sir.
Okay, thank you.
Yes, sir. Yes, sir. Yes, sir.
Okay, am I audible to all?
You're audible, sir. You're audible.
Okay. First of all, I would like to thank Dr. Nitin for inviting me for this FDP, Traditional and Artificial Intelligence-Based Training Program on Handling and Care of Laboratory Animals. It's a great theme, sir, you have chosen. And the topic which I'm going to present is record keeping for CPC SEA. Okay, may I know, sir, how many of faculty and students are there in this session?
Sir, the total registration we are having is about 300, okay. But right now, approximate 75, more than 75 faculty members in the meeting.
Right, that's great, sir. Because the topic which I am discussing is mainly for the faculty persons, as they are the record keeping bearer for CPC SEA. So I will, in this my presentation, the learner shall be able to know about the CPC SEAE, understand the objective and function of IAEC, learn about various records to be maintained by IAEC as well as Animal House, and understand the importance of record keeping. So this presentation is mainly important for the learner and the persons handling animal house facility, and the persons dealing with the animals for their research. So first of all, about the CPC SEA. So it's a committee, it's a statutory body, a committee for the purpose of control and supervision of experiment on animals. The short name is CPC SEA. It is a statutory body under central government that came into existence by the law Prevention and Cruelty of Animal Act 1960 under the Ministry of Environment and Forest. The main aim of this committee is to ensure that there no animal should be harmed during, before, during, and after the experiment on them. There is a huge debate between scientists and the social activists that persons handling animal like they are treating inhumane manner. So Menka Gandhi fought for this, and the CPCSE came in existence. The major functions of CPCSE comes registration and establishment of animal experimentation, of breeding of animal for this purpose. Means any experiment that has to be conducted on animals should be reported to the CPCSE. How they do this, we'll talk in the later. But for now, I'm briefly explaining what are the major functions of CPCSE. The registration, where the experiment will be conducted. The premise should be registered with CPCSE. This is the first function. Selection and appointment of nominee in the IAEC for registered establishment. If any establishment, organization, institute, university, any lab that have to conduct experiment on animals should have a institutional animal ethics committee, and there are few nominee. If you know the constitution of IAEC, the 5 internal member and 3 from CPCSE nominated by CPCSE. This is the constitution of IAEC. So the selection and appointment of nominee has to be done under CPCSE norms. Then approval of animal house facility for the basic report of inspection conducted by CPCSE. If a person has to conduct experiment, then the animal house facility should take prior approval from CPCSE. The approval based upon the location, the various guidelines that comes under CPCSE has inspected, and there is a separate performa for which we are going today. What are the various records that to be kept? Then permission of experiment involving involving use of animals. Means any animals you are using, you have to take permission prior from IAEC. That has to be permitted by CPCSE. Recommendation of import of animals from for the use of experiment. If you are using any transgenic animals or you have to import some other breed, then there is a prior approval needed. Action against establishment in case of violation of any legal norm or stipulation. If there is any irregularities in record keeping or the use of animal without prior approval, that the action that has to be conducted against them is governed by the CPC SEA. To achieve all these functions, CPCSE constituted institute level committee that I'm talking, IAEC. This is constituted under breeding of and experiment on animals, Rule 1998, and the later amendment come in 2001 and 2006. The timely, there are some amendments regarding the nominee profile, the selection, the fine imposed, and a change in the different parameters. So this all has to be done under the IAC. Means we can say if CPCSE is a center, then IAC is state, right? Under the constitution. I must appreciate if anyone interrupt or if any problem occurs, you can take in between the session. The main objective and function of IAC is similar to that of CPCSE. Means the prior function of CPCSE is executed under IAEC. Every experiment, every single experiment that has to be performed on animals should be under the supervision of a qualified person. This is the primary objective. Then experiment performed with due care of humanity as far as possible. Experiment involved are avoided, means animals, use of animals are avoided, as the theme of today's FDP is also artificial intelligence. How we can minimize the animal use, minimize the animal exposure for experimentation. Then experiment of large animal are avoided. It means where we can do the similar experiment on smaller animals like mice, rat, then we should avoid on experiment on large animal like dog and cats, right? Experiments are not performed merely for the purpose of acquiring skills. This IAC has, CPCSE has made amendments. Now, some of we know that we have used experiment in our UG and PG program for training purposes. But nowadays, we cannot use animal for training purposes. We only use animal for research purpose. So experiments are not performed merely for acquiring skills. Animal next objective is animals intended for the performance of experiment are properly looked after before and during the experiments. Means proper euthanasia, anesthesia, proper care, rehabilitation. This has to be taken care. And the last, suitable record are maintained with respect to experiment performed on animals. Record, the importance of record is must, because if you don't have record, you can, there is a penalty, there is a revocation of license, IEC. That it is another objective of IEC, that stable record has to be maintained on experiment that has to be performed on animals. The today's main topic, that is what records have to be maintained by CPCSE or animal house. Animal house facility is governed by IAEC to meet the regulatory guidelines laid down by CPCSE. Various record has to be maintained in the form of application forms, log register, stock register, etc. So these are the form of records that has to be maintained by animal house facility or IAC council. Record for IAC, these are the list of records that has to be maintained by animal house facility or IAC committee. The first one is curriculum vitae of all the members. As I told earlier, that IAC constitution 5 internal member and 3 external member. These member can be replaced, can be changed by prior informing to the CPCSE. So all the, if I told you the constitution, 1 biological scientist, 2 scientists from different biological stream, 1 veterinary, 1 scientist animal house in charge. Means these are the 5 internal person, but one institute or organization take head which is not a scientific person, then there may be 6 internal person, and 2 outside person means nominated by CPCSE or major CPCSE nominee, link nominee, scientist outside the institute, premise or university, and socially aware nominee. Means 8 person make the constitution of IAC, and for conducting any meeting of IAC, at least 6 should be there. If 6 person are not there, the meeting will, the quorum will not fulfill, and no meeting will be entertained, and the meeting will be postponed. If any person resigns from the IEC, then it has to be replaced by another person. So all the curriculum vitae that biodata of those IEC member has to be maintained under IEC. Copy of study protocols. Enclosed document progress reports shall be maintained by IEC. It means the other form that we apply, form B, for asking the animals, that has to, the record has to be maintained by IEC, whether it is selected or rejected. But if anyone apply for animals, then the record has to be maintained. Minutes of meeting duly signed by chairperson and all the members of committee will be maintained by IEC. For meetings, I must tell you that for any organization, it is mandatory to have two meetings in a session. One can do quarterly, semi-annual, or where animal use is less, one can have a meeting in a year. But ideally, there are two meetings. IAC meeting has to be conducted by member secretary of IAC. Where animal uses is large, one can conduct a meeting in every quarter, right? So minutes of meeting, in minutes, what agenda are put, whether the inspection of animal house is conducted, the all agenda has to be taken care, and the minutes of meeting has to be maintained. Copy of existing and relevant national and international guidelines. Means if there is any changes in the guidelines regarding means with what kind of anesthesia we can use, what kind of euthanasia technique we can use. Initially ketamine can be used, but nowadays it is banned. So those guidelines which are changing time by time has to be maintained by IAC. Then copy of all correspondence with members, researchers, regulatory body shall be maintained. Means if a person apply for research and its project get approved or rejected, then the correspondence copy that you have mailed them, you have initiated you have made contact with IAC or the member nominee, the all record has to be maintained. Project completion report of approved project. If project completion record, there is a separate performa, different forms I'm going to talk in the letter, that has to be kept by IEC. Record of breeding animals, supply, etc. Breeding of animal undertaken should be maintained. If a premise is licensed for breeding of animals, then record of breeding various species, the animal present in the animal house has to be maintained by IEC. Record of import of animal with species, source, quantity, uses. These, as I told you, there are various forms. So import or if you procure animals from somewhere, some other premise, the record has to be maintained in the form of Form C. I'm going to demonstrate in next slides. Record of all contact research. If a premise do contact research, like there are many organizations which do third party work, then they has to maintain their also record, whether you approved whether the performa is approved or not, whether IEC approved the performa, then all the record contact research has to be maintained by the IEC. Record of rehabilitation of large animal. If you use a large animal, then it is possible, if it is possible and where it is possible, the rehabilitation has to be done. So there are various methods of rehabilitation. I'm not going to talk about this rehabilitation right now, but the record of rehabilitation has to be maintained. All the document has to be archived for a period prescribed in SOP formulated by IAC. How many times you have to keep that record, that has to be finalized by the IAC, and the copy of same has to be communicated for CPCSE. In the time of RTI, one can ask how many experiment has performed in 2010. So you are not bound to tell if there are in meeting you have passed the performa that we will keep record for 5 years or for 2 years, for 1 year, for 10 years. So you are not bound to this if you formulate the SOP in IAC of institute. The purpose of IEC has to be done by animal house facility. All the things that IAC do with the animal house. So all these record has to be maintained in the animal house, but there are few addition that there should be present in the animal house. First is animal house plans, before the building plan, where is the quarantine area, where are different species has to be maintained, the washing area, the feed area, floor wise plan and fixtures that where these facilities are in animal house that has to be kept. uh, it is also required for applying for new registration in form. I will demonstrate in next. Animal house staff record. It both consists technical, non-technical. Means animal caretaker, the housekeeping staff, all the record has to be maintained. Daily diary of veterinarian. As I told IEC, there is a one veterinary person who look after the health of animals. So the daily diary of veterinarian has to be maintained. Feed and stock register, from where the feed is purchased, how many feed there in your animal house premise, all the record has to be maintained. SOP related experiments, care, breeding and management of animals has to be maintained. Breeding, stock, purchase and sale record in form of various forms like form C and form E has to be maintained. Again, minutes of IEC has to be maintained. Record of experiment conducted in the form of form D, that has to be kept. Mortality post-mortem record has to be maintained. Clinical record of sick animals is to be recorded. Training record of staff involved in animal activity. How many trainings IEC has conducted for the animal house staff, that has to be maintained. Water, feed and breeding material analysis report. As per the sanitation, the quality of water there in animal house, as per CPCSE guidelines, all the record has to be maintained by animal house. Health monitoring record of animals has to be maintained. And again, as I said, rehabilitation record wherever required, wherever you use the large animal, the record has to be maintained. Then come to the main part, that you now by now you know that what record you have to be maintained, that there are some pre-specified forms by CPC-SEAE. The record has to be maintained in different, these 5 type of forms are there. First is form A, that is for application for registration of with CPCI. If you are going to opt, if anyone from different institute, the people are from different institute and they are planning to open a new animal house for their organization, then they have to apply to CPC-SEAE in the form of Form A. There are different parts of this form, consisting from the location of animal house, the floor plan, as I told, the minutes of meeting, whether the IEC has consisted or not, and if consisted, who are the members of internal IEC member, what they have proposed, what is the purpose of opening the new animal house, whether it is for teaching purpose, for research purpose, for breeding purpose, all these has to be specified in Form A. Then come to Form B, submission of research protocol. If anyone wants to carry research on animals, then he or she has to submit the research protocol. The protocol guideline constitutes of the species, the age, the number, why these species is required, why this number is required, what is the age of animal needed, how you will going to perform those experiment, whether you anesthesia is required, whether you are going to do any kind of toxicity study, why this research is necessary. All these has to be submitted in the form of Form B by investigating scientist or the person who want to carry research. Then Form C, record of animal breed acquired, maintained by breeder or establishment. Means if there is a premise for breeder or establishment, then a person, if the premise is for breeding, then how many species are there, and the number of breeds, the number of animals in there, that record has to be maintained in specified per forma as prescribed in Form C. Record of animal acquired and experiment perform. If if you get approval from IAC and then you have to conduct the experiment, then during experiment, what kind of drugs you are giving, what anesthesia you are giving, how many animals you are sacrificing, the whole record has to be maintained by the investigator in the form of Form D. This is after you get approval of IAC and now animal are issued to you, then you have to maintain Form D. Then Form E, it is record of animal sold. This is for the breeder or where you get procure your animals. So the Form E is maintained by the breeder only, or but the rest form A, B, C, D has to be kept or maintained by all the animal house facilities. That's it. This is the performa Form A. As I told you, that Form A is for applying the new animal house facility. It consists detail of establishment, where you are going to, whether it is a government or private, because there is a separate fee for government and private, whether it is established under Ministry, Department, Council, or any other. This is the form detail for Form A. This is important for those who are willing to apply new animal house facility for their premise or department. Form B, I know most of the pharmacologist has applied this for their research. So you might be aware about Form B, and not only pharmacology people, other person doing research on animals and need animal for their research has already applied this Form B. It contains, as I told you, it contains primary. It has two sections. First section where the experiment will be conducted, whether it is a basic research or education research, contact research, from where you procure the animal, from where you kept those animal, where will be the where will be the experiment conducted. The section A consists of these things, but in section B, all the technical details, like as I told you, how you will going to anesthetize, how you will going to do experiment, why your research is so important that you use animal, why animal is necessary. To all those detail, if animal necessary, then what species, what kind of age, what will be the age of animals required. In toxicity studies, we write that you need mature. So what is the definition of that maturity? Some people say that 4 to 6 weeks, some people say 4 to 8 weeks. So why the IAC person pulls down the leg of pharmacology people on this ground? You write the mature, what is the definition of mature? So we are not trained for the anatomy of animals regarding the age, why it is required. We study from reference paper and write it, but IAC people ask about these. So in this consent, veterinary persons, help of veterinary person should be taken, and that what will be the identical feature of those animals, that is mature, juvenile. So this is the technical detail that has to be filled by the researcher in section 2. Then come to the Form C, means the record of animal breed or acquired. If you breed animal or you get it from any other establishment, then this record has to be maintained. Date of entry, the number of animal species, number of animal acquired, address and date from where, whom acquired, number of animal transferred, their details, all detail, the registration number of establishment, whom the animal are transferred, and the signature of animal house in charge. So this is the performa that has to be maintained for procurement. If you are not a breeder, then you have to maintain it if you are procuring from it, and if you are breeder, then it is also maintained. Then Form D, this is most important for a researcher. If your protocol get approved, then you have to maintain this record. After you get your animals, then this record has to be maintained day by day. date of entry, when you release your animals from animal house for research, the number of animals you are taking care, the registration from breeder, it is the bill copy is required. So you can't do experiment on another people. The project get approved for one person and the same animal is used for another. It is not so. Then date particular order grant permission of committee. When you get this approval, this IAC minutes has to be put, or the certificate there that this protocol is approved. To the date has to be mentioned. Period. The experiment in section B of form B, section 2, you have to write that how many time is required to conduct your experiment. If you don't issue your the animals during that specified period, then you are not entitled to get your animals. So name and address authorized for the experiment. The protocol get approved in the name of PI or the name of your guide, but the experiment is conducted by student. So here the name of a student has to be mentioned. Then certificate of investigator authority, whole experiment specified in has to be completed. Here your guide or the PI signature is required. Then this is the form E maintained by the breeder. Sold, number of species animal sold, whom you have said, date and IEC number, protocol against which you are selling the animals. So no breeder, no premise can sell animal to any other person. Like we get commodity, you cannot procure animals like it. For procuring animal, whether it is a independent research or for your institute, you have carry yourself with the IEC minutes and the total number of animals sanctioned. Then only you can get the animals for experiment. If any irregularities found in this, then there are penalty decided by CPCICAE. The next slide talk about the why record keeping is necessary. It is as mandatory guidelines specified by CPCIE. See, it is mandatory that we have to maintain all these record. Then record has to be produced as and when required during inspection by CPCIE for new or whether continuing the existing registration. So all record they can check. Irregularities in the records may lead to suspension or revocation of registration or closure of animal house facility. If those records are not properly maintained, or if any irregularity is found, then CPCSE nominee or the inspection committee may revoke or suspend the registration of that animal house or that IAEC. So that's why record has to be maintained. Thank you. I can see one question, sir. Why the animal should be quarantined? What is the purpose behind it? Okay, Radhika. Quarantine means acclimatize the animal. Means you procure animals from some other place that they have to be transported from one place to another. It is like we are changing place. We acclimatize ourselves for that particular environment. We think that we have to live here. So if you do not quarantine animal, there will be some biochemical changes, like stress may be high. So the purpose of your experiment will be failed. There is a false reading during the experiment if quarantine is not there. Some biochemical changes might happen if quarantine is not proper. And there is a proper guideline for quarantine also. You cannot quarantine two species together. You cannot quarantine. There is a particular space required for floor area for per animal. Transportation is different floor area, and for quarantine there should be a separate floor area, so that animal get comfortable for the place and you can do your experiment and get exact reading or proper reading. That's why quarantine is required. No, ministry for CPCC is same, environment, Ministry of Environment and Forest. Anyone else? Any query regarding for record or anything else you want to ask about IAC, Animal House or CPCC? Yeah,
सर एक क्वेश्चन
Dr.
है.
Yeah, just ma'am, I'm taking your question also. How to become CPCSE member? Yes, nice question. Dr. Deepak has asked. Dr. Deepak, I recommend you please visit the CPCSE website. There is a performa for CPCSE nominee. There is a you can. They have. I am asking. After 2018, they have changed that performa thrice, and now the current performa is there. They ask for various kind of details, like whether what is your qualification. Few people BSc, MSc in zoology or biotechnology, pharmacology people, they are entitled to get CPCSE nominee, and who has the research exposure of animal, they ask for it. Put your papers that you have conducted experiment and you might know about what is form B and how you carried that experiment. So it is required. And the back door for this is social aware nominee. Social aware nominee, if you are not from pharmacology background or from zoology persons, then the back door entry for like Shutix people is become social aware nominee. Be the person of like some social activities like PETA or something like that, and have some certification with you that you have conducted the or carried the awareness program for animal welfare. Then there is a backdoor entry other than pharmacology person to become a member of CPCSE. They get training time by time. They ask for nomination form. They choose from it on the basis of biodata, and they call for training, and you become a CPCSE nominee. Yes. Minimum number of IAEC, 5 people, integral, five from department, and 3 outside, social aware, 1 scientist from outside, and CPCA main nominee. If main nominee is not there, then link nominee. Minimum quorum for IAEC is 6 person. Out of 8, 6 should be present during the meeting. So this is the minimum quorum required for IAEC meeting. Out of 8 person, minimum 6 should be there. And in those minimum 6, the main CPCSE nominee or link nominee, and the presence of CPCSE nominees must for IAEC. No IAEC meeting is approved without presence of CPCSE nominee, main nominee or link nominee, right? If any failed experiment, then do we need to get permission again? Yes. That period, if the specified period of your experimentation is over, then you have to again take the permission. If your experiment fail, then you have give the particular washout time for your animals. If your protocol is approved for 1 year and you acquired animal in the initial the start of your tenure, and your experiment get failed, the number of animal you are asked are all are used, then you have to give washout time to remove the drug again, to maintain them, to avoid unnecessary exposure. And for that you have to maintain form D, that you have carried these experiment. The gap is given here, washout time, again you can use those animal for the same experiment. So no issue for that. Again you are asking for a specified period of time, you can use your animal again and again, but proper rehabilitation or wash out time after exposure, after the period is over, washout period is over, you can use your animal without any permission. How to become CPC? Dr. Deepak, I have answered. Raju sir, I have one question. Can you use animal at UG level for project work of? No. At UG level, no animal is allowed. Animal are allowed only for PG research. In PG program also, we cannot use for demonstration purpose. For those, the AI, the like FarmX software, the software, simulation software, animal simulator, these has to be applied, but not for the purpose of for teaching or acquiring skill. We cannot use. The IEC will not permit, CPCC will not grant. Only research, only for research. So we sometimes face some problem ki sir we have not used the animal, we have not handled the animal before, but at once how we can do to the main the guide can help out in that. Palvi said, sir, as you said, I is the state authority, so there is no role in central experiment. No, ये legislation वाला नहीं है. All IEC has to be regulated by CPCSE. This is not like independent state do whatever, but center is not interfering. Here, center full interference. And as I told, if record are not found proper, the revocation of license can be done. After experiment, yes, rehabilitation. We are not saying it rehabilitation. We say it like washout period. After 15 days is mentioning for washout period. Depend upon the drug you are using. Like for cancer, you cannot reuse again and again. Okay, anyone else? Why biotechnology? Yes, biotechnologies are there. And they can be the part of IEC. Biotechnology people can also be the part of IEC. As I told, science background scientist can be the part of IEC. Yes, anyone else?
Uh, is there any question from the side of participants? Please raise your hand if there is any question. Okay.
Shayad Salman Ali has asked if we want to carry the animal while traveling to other premise related to research work. Can we? Yes, you can do with the specified protocols as per CPCSE, like for traveling the animal, the what kind of vehicle you are using, the floor area should be proper for transportation, the proper feeding and water facilities should be there, air conditioned vehicle is required. So by these following these protocol, you can transfer your animal from one premise to other. Rehabilitation guideline for large animals, not for rodents or like it. It is a washout period and that's it. Yes. Yes, anyone else?
Anyone else? If there is any question, please you raise your hand. Sir, is it, sir, a question from the side of Rohish Pora?
Yeah.
Is it the responsibility of animal house in charge to maintain Form D?
Form B, to keep the record of Form B, not all the forms received by Animal House. Thus, Animal House in charge is mostly the General Secretary of IAC. So it's a responsibility of the person maintaining those record, who has applied and who get approval. So all these record has to be maintained. Yes, it's a responsibility of Animal House in charge, who have applied for who has asked for animal use, whose protocol has to be put before IAEC, and who get approval. This report has to be maintained. Yes.
So one more question by Dr. Priyanka. So what is Form D?
Form B, as I told, a Form B is a specified format for asking the animals required for your research purpose. So it has two sections. One section, whether from where you are acquiring animals, from where you carry experiments. The first section contains this detail, and the second part contains the technicalities of your about your research. Why animal is needed and needed? If animal required, then what which species is required? How many numbers? How is going to anesthetize? How is going to perform your experiment? How many models has to be carried out? That has to be given in section B, section 2 of Form B. So Form B is merely the form for asking the animals from IAEC for research. So it's a specified format. Yes. Form D is the responsibility of principal investigator.
Is there any more
Form
question?
D, Form D. There is a question from Dr. Priyanka that form responsibility to Form D, animal house in charge or principal. Principal investigator has to submit Form D, whether it is going to return or use consumption. A Form D is basically the consumption record that you animal issued to the PI. What the PI has done with the animals, that record is Form D. So for animal house in charge, Form D has to be taken from PI. Yes. Is there any more?
If any question from the side of participants, please, you can raise your hand. Sir, one more question. Rehabilitation is required for small animal.
No, it is for large animals, ma'am. For small animal, it is washout period, as I told earlier. There should be a gap after doing one experiment. If the animals are used for one experiment, or you can say that one model, that the another, whether it should be given proper washout period, means like 2 weeks, 3 weeks washout period for using them again for another experiment.
Okay. I think no more question. Okay. Sir, now there is no more question.
Okay, ma'am.
Okay. Thank you so much, sir. Thank you so much, sir, for such a informative and motivational session. And thank you so much from the side of all the participants to answering all the questions in a very easy way. Thank you so much, sir, for cooperation. Thank you so
Thank
much,
you,
sir,
ma'am.
from the side of MIET. Thank you, sir.
Thank you, ma'am. Thank you all the
Thank you,
participants.
Shivkant sir, for such a wonderful session, because thank you, sir, for such a wonderful session, because being a faculty member, we are unaware of means what kind of the forms or the documents we have to maintain while we are conducting our animal experiment. So this was very informative sessions, and I hope all the participants will get benefited by this lecture, and surely they will maintain all kind of records, all kind of documents while they are conducting their animal experiments. So thank you once again, Shivkant sir, for joining us.
Thank you, Nitin sir, for inviting me. Thank you all the participants for listening, and thank you MIET. Thank you.
Yes, sir. Okay. Our next speaker of the day, Dr. Vikash Medi, sir, has joined us for the next session. Thank you, sir, for coming. I welcome you on behalf of MIET. Thank you, sir, for joining us. I would like to introduce our second speaker of the day. Sir, am I audible to you?
Yeah, I can hear you.
Oh, okay, sir. Thank you,
Yeah.
sir. Okay. Dr. Bikash Medi is professor and ex-additional medical superintendent in PGIMER, Department of Pharmacology. His area or expertise is experimental pharmacology, clinical research, regulatory pharmacology, development of nano formulations, pharmacogenetics, pharmacogenomics, and stem cells, etc. He has more than 25 years of teaching and research experience as more than 65 prestigious national and international awards, namely Dr. D.N. Prasad Memorial Award with Gold Medal 2009 from Indian Council of Medical Research, New Delhi, V.K. Bhargav Award with a Gold Medal from National Academy of Medical Science 2013, R.N. Chopra Oration 2014 by Indian Pharmacology Society, Anand Datta Award 2016 by Indian Pharmacology Society, Dr. B.N. Ghosh Oration at 43rd Annual Conference of IPS and International Conference on Pharmacology and Translational Research. He is in core panel of experts for task force in IND application and regulatory affairs. He has lead GLP inspectors, GCP inspector, principal assessor for NABH. He is in several committee in DCGI office, ICMR, DBT, DST, CSIR, NBE, MCI, etc. He has authorized 5 books and 150 chapters and published more than 400 articles in national and international journals. He was selected as a member of various central government committees and central government funding agency in the IND committees, FDC committees, compensation committee, subcommittee of drug regulation amendment committee, central working group committee for pharmacovigilance, Signal Preview Committee, member of Biological Task Force Committee, member of GCP Inspection Team, DCGI, Core Training Panel Committee, Pharmacovigilance, National Formulary Committee, IPC, Chairman Selection Committee for TA, Special Invitee for National Advisory Award of Homovigilance Program, Special Invitee for Central Ethics Committee, ICMR, member of the Committee for Evaluation of Spurious Drugs, Antimicrobial Evaluation Committee, Basic Medical Science Project Evaluation Committee, at CG Vaccine Committee, member of Technical Committee for ICMR Institute, core member of Committee ICMR, member of ICMR funding to CDRI, special invitee TB Task Force, DBT Task Force member, the expert member for anti-doping and member of expert group NEDA, National Board Examination, Sanity and Selection Committee member for NIPER, member of Board of Study of Punjab University, Chandigarh, working as GLP. I would like to welcome Sir. I think there is a very short word to introduce to. I would like to welcome as a guest of the day on behalf of the Department of Pharmaceutical MIT Technology. Please welcome, sir.
Yeah. Thank you, madam, for nice introduction. So I just request you to make me co-host so that I can upload the slide. Can you make me co-host for uploading the slide, please? Can you hear me?
Vishwashi, can you help us to join the Vishwas sir as a co-host? Vishwashi?
Vishwas, sir.
Because sir, please wait for the moment. I am calling to Vishwashi and he will lead you to co-host.
You have to make me co-host, then only I can upload the slide. Yeah. Yeah. So can you see my slide?
Yes, sir. Your slide is
Nitin?
visible.
And I'll just check it. Is it moving, the slide? Is the slide is moving?
You can continue, sir.
It is moving. Audio is okay, no?
Yes, sir.
Yes, sir.
Okay. Thank you. Thank you. So thank you for the opportunity. I like to thank organizer for inviting me for this lecture. So as since yesterday I could see the program, and all detailed animal experimentation has been already been discussed. So I'll be giving the briefing about that current regulation and special reference to how to validate the animal model. I'll just take an example of some of the neurological disease, and also for student purpose that how you should do the regulatory toxicity study when you put in drug development process. So if I begin that, though you might be thinking that there are so many drugs and drug for medicine for all the disease, but if you quietly, go into more specific one, that there are so many diseases we do not have not a single medicine. And that is why the new generation should be trained that they should be pinpointed at this level only, that you have another 30 years. Why not to take one of this, agenda, so that you can think of some of the molecules which should come to the market. So you can take an example of NOT, especially we get a lot of celiac disease patient. And if you look at this, celiac disease, we do not have any medicine. So that is one example. Then autism, you have several examples. As you know that earlier, if you see estimated 2017, that there is a huge, budget is required for the medicine also for development. But at least you can repurpose the drug that we learn it during the pandemic situation. So just in a brief that we go for a preclinical testing, especially in laboratory animal. Before that, we do a lot of, studies like in silico study, in vitro studies. But ultimately, if you see that all these studies, what we do, we actually like to see that how safe it is compound and can you estimate or evaluate the biological activity. So even if you go for 5,000 to 10,000 compounds, so ultimately, the success rate it may be 5 to 10 compounds. So, and it is going to take around 10 to 12 years also. Now, grow gradually, like once you do in vitro study, before that in silico study, then you go for, regulatory toxicity studies, that animal toxicity study, I'll talk about detail. Then you go for the different phases, like in phase 1, phase 2, phase 3. And this is also going to go for around 5 to 8 years time, and ultimately you file an IND. Now, altogether, if you see that regulatory approval, it is also going to take 2.5 years. And then you go for a phase 4 study. So this is all, talk about how the drug development process is going on. But as you see, recently we learned a lot of new experience in pandemic situation. So initially, the conventional technique is that you synthesize or purify the molecule based on in silico or in vitro. Then you go for a short-term animal toxicity study. Then you go for a long-term animal toxicity study, depending on a clinical use. Suppose if you want to use a drug for diabetic mellitus, where you have to go for more than 6 months or years together, so you have to generate a long-term toxicity study. Similarly, as you can see, we go conventionally that phase 1 study, it is in a healthy volunteers. Unless there is you have an anti-cancer drug which can cause cancer, you go for a patient. Otherwise, you go for a patient in phase 2 study, then larger patient in phase 3 study. But as you learn during pandemic that there are 250 plus drug has been trying to repurpose for corona. So this is also in that situation, you have an accelerated development review. So all the drug, if you see that they have applied to the regulatory authority and they got an approval as a investigational new drug. So this is also one of these days that how in emergency situation you can repurpose the drug. So these are the process. So when you do all the animal experimentation, you make an application, you call it IND, investigational new drug. So after doing phase 1, phase 2, phase 3, once you complete phase 3 trial, then you apply for NDA. NDA means new drug application. So after NDA, you get an approval for marketing authorization. So we call it a new drug till in a market up to 0 day where it is marketed until 4 years. So every 6 monthly, they have to collect the information about safety information and submit to regulatory authority. So this is what the phase 4 we do it. Now, coming to the current regulation, as you see that we are regulated by CPC SCA. And a lot of students in the exam, if you ask what is CPC SCA or CDSU, they'll not able to tell it abbreviation. Now, look at the guideline from the national, perspective. We have CPC SCA guideline, and I just wanted to make it clarify one thing. It's also normally when you say guideline, guideline means you may follow or you may not follow. But here CPCA guideline is attached to Animal Cruelty Act 1960. So it is a mandatory thing that you have to follow the guideline. Then we have a guideline from ICMR, guideline for laboratory animal. There are a couple of guidelines from ICMR. Then we have a guideline from INSA also. And also similar preclinical guideline, biologics for India also. Then we have an Institute of Biosafety guideline, WHO guideline. And similarly, international guideline also, those who are doing a contract research organization, they do animal toxicity study. So they have to follow that all the, like people coming from USFDA, EMA, they come for inspection. So they have follow all the international guidelines. So we have series of international guidelines from Japan. Then we have a guideline from Swiss guidelines. So all these guidelines they follow. So we have a national guideline, but mostly we are concentrating on CPC SCA guideline. Okay, so we have guideline from INSA also. So these are the guidelines. Now, as I think the earlier speaker had already talked about that CPC SCA guideline. So this is one important that before you, like you happen to go to any lab outside, even if you are a professor, you have been working for last 20 years or 25 years, still, they have to have they have an SOP before you enter into animal house or a lab, you have to go for an orientation program. So normally if you see that recently that we do not have much, if you ask a question, how many facilities we have a monkeys in India, you can just count it in a finger, because it has reduced to such a number that we do not have. How many facilities we have a big L dogs, you can count the numbers, because we had a GLP inspector, we know, because we go to almost all the lab. So otherwise, if you see that students should know that what are the smaller animals, how do you say it is mammal? So commonly, if you see that most of this experiment, we do it in, mice. We have, basically, if you ask the question that what is the species of the animal, so they are not able to identify or tell in an exam. So commonly we have a Wooster rat or Sprague-Blue rat. So we have a different but common animal, like suppose if you say Swiss albino mice, or if you go for a pharmacokinetic studies or keen sensitivity studies, like we have a rabbit and we have a guinea pig. So these are, animals are already available. Now, question here is, there are a lot of, discussion is that do you have a right to do an experiment? So here, if you see that as per CPC SCA guideline, we have to mandate that how should be the animal house. It has been already discussed, all the record maintenance. So if you have a proper area for, keeping this animal, definitely it is allowed to do an experiment. Of course, we can have that a couple of years back that there was a recommended that there will be no experimentation for practical purpose. Then again, it has come back to normal one. So as I said, the guideline, you can just go through the national guideline and international guideline, and that will give benefit to each and every point. You can discuss that what are the principles. Now, if you see the guideline, basically guideline, the CPC is a guideline, which is Committee for Purpose of Control and Supervision of Experiment on Animals. Just look at that, what is the motto of the guideline. Basically, it promotes that whatever experiment you do, like when our patient comes to the hospital, even if you happen to work in Western setup, for examination, you say, can I examine you? I have to examine, you take a history. So these are information, like you take a consent, maybe verbal consent. Similarly, whatever treatment and management you do for human, it should be promote for animal also. So basically, it talk about how to enhance the animal well-being and also quality. Like if you happen to go that you have an animal, okay, every day veterinarian, they check for their clinical examination or basically examination whether it is healthy animal or not. So that is how it is promoted as per CPCA guideline. So basic function, as I say, that there is a accountability of all the animal facilities for registration or selection or appointment of a register. So it is all regulated. So how many, so there is a timeline for this also. They come for a surveillance inspection also, they approve. So any animal ethics committee also, we have a mains CPCSA nominee or alternative one. So altogether, if you see that it talk about, keeping the record, which is we are very poor. If you ask the animal house that how many SOPs you have, they do not also know how to write an SOP and what is an SOP for SOP. So this also, what is a control SOP, what is an uncontrolled SOP. So all the record keeping and whatever you do, because today you are working in animal house or some experiment, tomorrow somebody else is come. So even SOP that there is a uniformity and you ask for a, reproducibility, veterinary care, animal, how do you procure the animal? Like, that if you say that you have a vendor, established vendor, which is approved vendor, they certified about the animal, then quarantine, stabilization, separation, these are, it should be included in SOP. How you go for a surveillance, diagnosis, and treatment and control of disease. So that is why that moment you, procure the animal, what you do is you have a room where you have a keep the surveillance, especially suppose hire animal. So you observe for, are they having, suppose you have a dog or you have a monkey. Monkeys are more susceptible for tuberculosis. Similarly, dog and cat, it is susceptible for rabies. So initially you screen the animal. That is what, that all the animal care and all the technical person who are experienced on doing that. So everything you look at that. So normally when you say, earlier it was Ministry of Environment and Forest, now it is going to the Animal Husbandry Division, the CPCSA. So under this, that we have an Institute Animal Ethics Committee and we have a Institute Biosafety Committee. This is required because any hazardous chemical you wanted to work. So initially you have to clear it from, Institute Biosafety Committee. Suppose you want to work on, PTZ, for example, common example. So PTZ could be accidental, ingestion or injectable. It could cause a seizure. There could be mortality. So do you have an antidote? How we are going to keep it? So you should have an SOP. Similarly, for radiological active material, there is a particular procedure for disposal that you have to liquefy the animal. And how do you dispose the biomedical waste? So once you clear that it is not hazardous, then it comes to Animal Ethics Committee for an approval. Similarly, we have been working for last 10 years. There are, stem cell. So in case of a stem cell, before you apply to Institute Animal Ethics Committee, it has to be cleared from stem cell, committee. So once it is cleared, then it comes to Institute Animal Ethics Committee, then you get an approval and you can do the experiment. So there is a provision for a member for this that include biological scientists, 2 scientists from different biological division, then scientists in charge of animal house. So this provision is there as per CPCA nominee, and you have to keep all the records. Similarly for IBC also, Institute Biosafety also, we have the head of the institute and his nominee, or 3 more scientists who are basically working on DNA or molecular biology. And also we need a 1 person which is qualified person at the biosafety, which have been, background of radio, you have an experience. So 1 more member from DBT also. So these are compositions are there. So this is a regulation. Every institute should have a biosafety committee. We should have a stem cell research committee, then Institute Animal Ethics Committee. So these are some of these, the head of the institute and nominee. How do you do it for IBC? I have already talked about, and similarly for Institute Ethics Committee, you have a chairman. But here it is, that 1 veterinarian doctor who is looking after the animal house is also a member, and animal house in charge also a member. And you have an outside scientist also, socially, active members is also there in this case. So when you do animal experimentation, there are principles. Basically, what I said about goal about that you have a human treatment to the animals. Okay, so basically that if you have some alternative method, you are going to replace the alternative method that we have seen in cosmetic. If you see, there are so many tests it is not allowed because there is an alternative experiment is there where you need not have to use the animal. Then they reduce the number. So whatever the number statistically it is justified, like you say one group, maybe 6 animal is required. But if you go for a surgical, procedure, there could be mortality. So you have to keep it around 10 animal, because if you find that there are 10% mortality, suppose you work on traumatic, uh, brain injury. So there you have to go for craniotomy. Maybe mortality is high, so accordingly you have to keep it. So whatever number is there, then please go to the ARRIVE guideline also. I'm not talking about international one, ARRIVE guideline. It talk about that you need to justify. If it is 6 or you need more 8, then you need to justify, because then refinement. Like we do that surgery, so we take an injection. So that this is a painful procedure. You give an anesthesia. So refinement that it should cause the minimum pain, asthma or injury to the, Then fourth, rehabilitation. I think people have asked the question that rehabilitation, even a smaller animal, we are going to rehabilitate. Like for example, we do a pharmacokinetic study in rabbit. You are not going to sacrifice the rabbit, you're going for a rehabilitation. So similarly for higher animal also. So for both animal, it is rehabilitation procedure, fourth is data. Then fifth, you can take it as we put it in my book, is responsibility. So we have a replacement, reduction, refinement, rehabilitation, and we started one of these art is responsibility as an investigator or as a researcher. So there could be alternative to animal experiment, as I refer it to cosmetic drug and cosmetic tag. We have a lot of cell culture in vitro studies where it can be replaced, and you need not have to go for in vivo. But one thing is that when you talk about the medicine in the drug therapy, you always say that there is an individual variation. Then you also say there is an intra-individual variation also. So none of this method, maybe you can have an alternative. It is going to, extrapolate the data that how much intra-individual variation or inter-individual variation is there. So this is only you can see the biological variation by doing this. But at least you can have a rough idea that if you do in a in vitro studies or non-living system or simulation, because when you take a evidence, in a simulation, they will take a no evidence, but at least you have a rough idea which could be a potential molecule for that. So we have series of alternative to animal experiment, but it cannot be replaced the whole animal. So CPCSA guideline, it talk about care and handling, but one thing is that most of the speaker, what I could see that these are, including me, we have a GLP inspector. So there should be have been a class for GLP also, especially OECD GLP. So this could have been next maybe program we can include a lecture subsequently. Now here it is one more point is that when we happen to go to any lab, whatever we ask that, that all this, if you take a mice, rat, hamster, guinea pig, rabbit, cat, or higher animal like monkey and dog, so they have a specific profile. Like if you say, I go for hemoglobin, my hemoglobin is average on an average in Indian population is 12.5 to 14. Okay, male and female, there is a difference. Now, what about the animal? So when you go to lab, if you ask that you are doing a hematological studies, and suppose the DLC, differential leukocyte count, what is your reference standard? So you might get a answer is a human standard. So that is what that all the lab, when you do a preclinical toxicity study or animal study, it should always refer to the historical control. Like you have been doing for last 10 years, what about your historical control or reference standard, because you're referring to the human standard. So this is what need to be clarified in animal studies. So similarly for any different protein, albumin, whatever blood biochemistry, what do you do? It should be referring to historical control for last 10 years, you should have a data. Some of the word people do not understand, as I said, that if you ask a student, what is the species of the animal? They're not able to understand. How do you say it is pure breed, pedigree, crossbreed, mixed breed? Because moment you say Swiss albino mice or rat, albino rat, so you say specifically say, because most of this animal house, they do not maintain the breed. So it is a mixed breed. So that can be looked at. Now, as I say that most of the university, when you go, they will also tell you about the guideline as well as that, physical skill for handling of the animal. Because that when you give an animal and you see if it is aggressive, you get to know that these animals are never handled. Okay, then you look at the tail of the animal, you can get an idea that how about the maintenance of the animal. If the tail is dirty, you get to know that they have not done maintenance as well. So that is a rough idea to get it. So this is where that the most less pain sensitive, if you see the animal, if you put it in nape of the neck. So basic idea that when you hold the animal, you just grab it, because animal will bite if you allow to turn the head, or if you are transporting from one place to another, you just take a blunt forceps and like that you can go. So you should have a technique for how do animals. So this is one picture that if you grabbed it, you know, slightly, so you're not allowing the animal to move, but they will be very irritable if they are not able to breathe it. And that is why that you have to just, when you hold it, it has to be very clear that you are allowing the animal to breathe normally and there is no pain, less pain. So now some of the animal, like you take an example of rabbit, very, very docile. So you take it in a flank, like I have hold it in a flank. So there is no problem. They'll be very docile. Similarly for guinea pig also, very, very docile. So you hold animal on the flanks. So that will have, then you then experience that there is no problem for, handling the animal. So these are some of these housing requirement for animal. So every animal as per CPCSA guideline, so what is the case, how do you maintain it according to body weight, how many animal you can keep it, like for in case of a mouse, rat, or in case of a higher animal. So these are there. Now food and water, you know that you happen to go to any GLP facilities, they'll say that do you have a certificate for water certification, because RO system should be there, and at the regular interval you need a certification. Similarly for any animal food, when you procure, they give a certificate, and randomly also batch you send for analysis to a government lab, that what is the constitute of the particular animal food. So that also all the records should be there. So these are physiological one I have already discussed, I will not go in detail. So any of the procedure, if you see the holding that physical restrainer, so these are some of these animal that when you approve that it is allowed that it should be less than 3 years. And also you should know that that normally when you examine the animal, that you have a clinical sign. How do you say it is a healthy animal? So you have a clinical sign. Suppose you say that animal is growing at a normal rate. Suppose you include in an experiment 4 to 6 weeks that how it is growing. So you have a normal curve. Then what about the activity A to Z? So total, if you see clinical sign, there are 27 clinical sign, and that you can record it also. So if there is any toxicity, or normally that if you fall sick, what you do? You do not like to eat. Drinking is less, water drinking is less. Similarly for animal also, if there is a toxicity, then they'll not take the normal food and water. So that will indirectly signify that there is some problem. Or if you find there is, loss of fur, so that it will signify there could be a toxicity. Then you see all the orifices. Is there any bleeding? So like that you go with appearance and behavior, body condition. So any of the red tears, definitely there is a problem. So that's how you examine the animal. Now, when you do behavioral testing, I'll talk about detail. This is very important that you keep the animal into the place where there is a no noise, less dim light is there, and accordingly you arrange for any of the behavioral testing. So all CPCSA guideline, it talk about anesthesia. I'll talk about like earlier when you used to do a dog experiment and cat experiment. Suppose blood pressure regulation. We used to, record a mean arterial blood pressure where you cannulate the carotid. So there, if you see the anesthetic agent we use is chlorolose. It is hardly you find it today, because if you ask that why do you select the chlorolose, reason behind that chlorolose, it does not interfere with the reflexes, and that is why it was selected. Similarly, suppose you want to do a ECG, you are not going to use the ether. Ether is also banned as per CPCSA guideline. Why you do not use ether? The reason behind, if you use ether, there could be cardiac arrhythmia. So similarly for euthanasia, also there is a particular procedure, physical method, or you have an anesthetic gas chambers where you give a carbon dioxide and it is a painless death. So this is what, like you might have seen the euthanasia used by some of this legal proceeding for people who have a long standing. You remember that one of this patient was there for decades together in a particular Bombay hospital. So this is also in animal we have an euthanasia. So these are recommended one, what are the anesthetic agent can be used, but you have to select that what is the duration of procedure. Suppose you're a new person, you wanted to do a stroke model where you want to click the middle cerebral artery. How much time is required? Then how much duration of anesthetic agent? So depending on that, you select it. So these are method for euthanasia. I'll not go in detail. And physical method is also available. So these are method is approved by this. So route of administration. This is very important. One of the point I'd like to highlight. Please select particular animal, and if you are giving a route of administration orally, which is most convenient, that how much capacity is there. You have to also think that animal is also drinking, taking food at libitum. So how much capacity is there? Normally you just calculate it. Second point I'd like to highlight is when you have a plan for 15-day studies. So you recruited 4 weeks of the animal, and accordingly it is growing also. So normally what mistake it is done is you calculate the dose, then same dose is given. So you need to also see that animal is also growing. So if you say calculate the dose for per kg body weight or 100 grams, or what is the increase is there over a period of 10 days, 1 week. So this accordingly, so you select the IP route or oral route. So there is an SOP for that. How do you give an intramuscular? What are the site of this? So these are intramuscular or subcutaneous, because subcutaneous also you need to have a space. So normally we select if there is a large volume is required, you have to give it through nape of the neck. So you have a large volume because you want that drug to be absorbed, because there is a lymphatic circulation is there. So that's how we select the dose, how it is given orally or intravenously. We have a tail vein. If you see the tail, there is a middle vein is there, and there is a lateral vein is also there. So normally how much you can inject into the tail vein, so you calculate that. So when you withdraw, then vein will get collapsed. So these are practical skill is required for this collection of from the tail vein also. Normally when you collect the vein, because nowadays retroorbital it is not allowed. So what you do is you just cut the tip of the tail, and as we have a very sophisticated analytical method, so even minimum blood is, you can do the experiment. So these are some of this volume which is recommended. How much, like normally when you go for a blood donation, how much blood you give? You give 300 ml. So you have a 70 kg man with 5 liters of blood. So we allow to 300 ml. So that means you can percentage that you have a 250 grams of rat, or 150 grams of mice, how much it can be allowed. So generally it is safe to have 10%, but it's not beyond that. So this is the ethical angle, that how much it is allowed, or especially in pharmacokinetic studies where you have a repeated blood collection. So there you can design a study that 1 point, maybe 3 animal you sacrifice, you take out the blood if you need more. So like that also you can go for it. Now, this is difference between a baby rat and a mouse. This is very important. Normally people ask that you are going to include what is the species. I say that what kind of rat, is it SD rat or is it twister rat? How do you differentiate? Suppose you have a small rat, baby rat, or you have a mice. How do you differentiate it is rat or mice? Or how do you differentiate between male and female? This is a common question people cannot answer. So please go through this. So this is determination of sex. This is very important. Especially I'll talk about working on autism, where we look for a male pups, because male pups are more susceptible. So there initially during the pups you have to identify whether it is male or it is female. So there is a particular criteria where you can identify male and female rat. So these are the point need to be looked at, and then you can do. Anesthesia I have already talked about, and guideline I have already talked about, so I'll not go in detail. So these are point. But ultimately, if you see that, when you do an animal experimentation, we have to see that every animal has a right to life, and we should not take it unless we justify. That is why in a Form B, when you see that, is it done earlier, or this is a new experiment, it has never been done before, and you have a justification that it is in silico done, in vitro done. Now I want to see in vivo. So that is why you give a justification for this. Now I'll take you through some of the in vivo experiment, like in pharmacology and other discipline also. whenever we think of a drug, we get to know that, okay, what is the route of administration? So if it is oral or IV or any of the route, then we want to see that how pharmacokinetically it is there. Then correlating with a pharmacodynamic kinetic, we also go for a pharmacodynamic action. So basically, we want to reduce variability. So these are experiment it is done. So while doing the experiment, it is very important to calculate the dose. Now, if you take an example that how much dose is required for a pediatric newborn or adult, we always calculate the pediatric. And you might be thinking that pediatric it is lesser dose, but it is not lesser dose because the dose is depend on the metabolic rate of the any of the living person. So you take an example of the mice, rat, or a guinea pig or hamster. So comparatively, if you calculate the dose is more in case of a mice compared to the rat. More the smaller. I can give you an example. Suppose you take an example of elephant. Elephant heartbeat is maybe 18 to 20. But if you take a small bird sparrow, his heartbeat is more than 1,000. Similarly, for mice, heartbeat is 450. Rat is 250. So this is correlated. So what do you taught in yoga that you try to control the respiratory rate, you try to control the heartbeat. So that is why. So more the metabolic rate, you require more drug. And that is why the conversion factor, if you see that if you are giving a known dose of a human, which is, particular, milligram, so you multiply it by conversion factors. For example, mice conversion factor, you can see it is 0.0026. So if you convert into it, then exactly it will give the drug required for 20 gram of mice. Similarly, for rat is 0.018. And that's how you convert. So you can calculate the dose from in vitro study, extrapolate the dose, what should be the dose for, in vivo. So normally when you extrapolate, you take a median dose, then you take a lower dose and higher dose. And that's how you do a pilot study. Now, some of these experiment, what we do in behavioral testing, as I say that behavioral testing, you have to be careful about the noise, about the light, then animals should be familiar in that place. That is why that when we procure the animal, we wait so that it get acclimatized. Now, if you talk about, learning memory, so whenever you do a behavioral test, it is always then a battery of tests. Because if you give a drug, you say that what is the effect on a drug? Suppose you say that any drug which is have a CNS depression activity. So you can just do it. You give the drug and see you go for a writing reflex. Now, if it is you are doing a writing reflex, that means animal is not able to correct his normal posture. So what you get, so you have a rough idea that maybe it has a CNS depression activity. Then you think of that memory, short-term memory, long-term memory, how it is going to affect the coordination. Is it affecting the cerebellum? So you have a rotarod test or the grip test. So you have a several, battery of tests you have to do it. Nowadays we use in a neurobehavioral lab, we have a neurobehavioral lab. Suppose you do a Morris Water. So you train the animal. Okay, then you have a software because you have to track down all this, how he learned it, because basically how it is moving. So normally if you give, is it going to the right direction or whether animal has learned or not. So everything. So you have a control animal, we have a treated animal. Like that you can do an experiment. Similarly for rotarod also, you have a mice, you have a rat, you have a rotarod. So basically you wanted to go for a coordination. So any drug, if you see that, is it affecting the coordination? So there could be muscle relaxation also. It is not going to have a CNS depression activity, but indirectly you say that it is affecting the coordination because of muscle relaxation. So anti-anxiety drug, we have an elevated plasmase. So you can record all the movement. So normally when you see the animal behavior, that it is going to go because when there is a height, suppose you are standing on a building, what is your anxiety? Anxiety is maybe I'll fall. So you have an anxiousness. Similarly, when you put in an elevated plasmase, it is in height. So animal try to go to the darker area. It is going to go to the darker area. So if you have an anxiolytic drug, so it is going to, so you calculate from this, so any rough idea that how the drug could be anti-anxiety drug also. So we have several examples like catalepsy. If you take an example, like you might have seen that, psychotic patient where they become very aggressive. So one of the drug in acute emergency we give is haloperidol. Similarly, if you take the example that this is a rat and we have given a haloperidol, see the animal is still sustained in that position. So that is why we try to come the clinically correlate that if you give it any psychotic patient where it is very aggressive, you cannot maintain it, and then you give a haloperidol, it is in a similar sustained condition. So this is what we call it catalepsy. Now, some of the model you can see that you can work on cerebral ischemia, we can work on Alzheimer's, Parkinsonism, several models. Because none of this drug, we took an example of epilepsy also, none of the drug we have a curative. If we take an example of epilepsy, maybe there are 60, 70 drugs, but none of the drug is a curative drug. So we have still that we do not have a medicine for anti-epileptogenesis. Only one minimal effect is maybe valproic acid. So we have number of examples. Now, if you see cerebral ischemia, that you can induce the model by blocking this or putting a clamp on common carotid artery. So when you experimental language, if you say that are you going to give for universal ischemia, so that is you call it global cerebral ischemia. You can only produce global cerebral ischemia if you block both common carotid artery, or because 10% of this blood supply it is done from the vertebral artery. So if you do blocking the common carotid artery and the vertebral artery, it might causes global cerebral ischemia. But normally what we do, you block the common carotid artery, we say global, it is not global cerebral ischemia. Only one of the model where you can use Mongolian Jebdils, where you can use by blocking that common carotid artery, you can generalize cerebral ischemia. So here it is very important because in a Mongolian Jebdils, they do not have vertebral arteries. So if you wanted to clinically correlate, ideal model we have is middle cerebral artery occlusion, where we put the filament and we block the artery. But that way, if you see that many of the patients we have a ischemic stroke, like we have a transient ischemic attack. Many of these we have a thrombus, where it goes to the circulation we call it embolus. So there is because of embolism, it blocked the artery. So you have to clinically correlate all these aspects. So this is a procedure that if you are going for a bilateral common carotid artery, given anesthesia, then you isolate. So whatever. So when you will plan for an experiment, you have to plan it that what about pre-operative care? What about during the operation is care? What about post-operative care? So that way you have to plan it and you have to do it. So all this procedure care, you'll ask for it. So this is a procedure you can do. You can also go for a video. We have some of the videos is there. Similarly for Alzheimer's also, that we have an in vitro one, because basically you wanted to see that is the drug is acting through acetylcholinesterase activity. Many of the drug we have, donepezil, galantamines, many drugs are there. However, when you plan for any experiment, it is very important to also estimate the pseudocholinesterase, because that way is its lab. Maybe it is acting on acetylcholinesterase, but we do not have a data for pseudocholinesterase. So when you send the paper, the editor might ask that what about effect on pseudocholinesterase? So you can plan for in vitro study, or then you also think of in vivo studies, where that thinking of in vivo study, there are so many hypotheses. Now, earlier, if you see the Alzheimer's disease, people think of that it is because of accumulation of beta amyloid. Then later, if a couple of years back, people are more discussing about the tau protein. Now people are linking with diabetic mellitus. So if you think that it is because of beta amyloid, you have a model that you can, stereotactically, like you put it into the lateral ventricle, beta amyloid. So it will get circulated, because this is a degenerative, like it is long-standing disease. It is only, developed after 65 years or 70 years. So here is the procedure that you directly put into the brain in the lateral ventricle. So it is circulated. So this is linking to the beta amyloid. But if you see it is linking to the tau protein, then you can put the drug called isoprotenolol. Similarly, for diabetic mellitus linking, then you put the streptozotocin. So this is where we have to do. Then again, you do all these behavioral parameters and also molecular parameters. So that's how you can validate the model also in your lab. And these are experiments should be done, behavioral. I'll not go in detail, and all this. But single parameters, it may not be, going to establish this. That is what. So all memory tests, the rotarod coordinations, all these parameter you need to do it for special memory. Similarly, for anti-Parkinsonism also, we have an INV model. So most of the established model is MPTP model. So if you are going for a do it, because every model in experimental setup, when you discuss, because I'll talk about how do you validate the model also. So you have to also think of what are the limitation of the model. Then you can modify this particular model also. So when you modify it, then you can see that how you're going to validate when you modify it. So these are some of the common that how we are going for Parkinsonism disease. Now here, one more important point I'd like to highlight, that people, when they plan for an experiment, please remember that you have to also see that how the drug is given orally, intravenously, okay, IP, but at the same time, what about the solubility? Because sometimes people give the drug which is not soluble, and you have to establish the pharmacokinetic procedure also. So is it soluble in water? Is it soluble in DMSO? So suppose you plan for a neurological disease, and drug is soluble in DMSO. So when you send a paper, they'll ask you that you didn't keep a vehicle control group with DMSO, and DMSO itself is a neuroprotective effect. And that is why initially when you write the protocol, please also include another group of DMSO group. I'm just giving the example so that you can justify that it is also neuroprotective, but you have a separate group, have a comparison. So this is what the solubility you need to look at. Then you can just go through that what is about solubility, where it is soluble, how much it is soluble. So everything you can go for it. So this is our definition. Now coming to common experiment, what you do is recording the blood pressure. So normally in student, we wanted to know that whether people know how blood pressure is regulated. These are a common question for exam. Now if you see how do you record the blood pressure, you just remember to Dell. Dell is from Cambridge. We have a Dell spectrometer phenomena. So if you look at the history of the Dell will tell that recording of the blood pressure is a direct measurement as well as indirect measurement. What he used to do is indirectly, he used to put the cannula and record the blood pressure. So if you today see how to define the blood pressure, it is a lateral pressure. You put a cuff, you measure the systolic and diastolic. But in animal experiment, when you measure it by putting a cannula, you're not recording a diastolic or systolic. You are recording a mean arterial blood pressure. So how do you define a mean arterial blood pressure? So normally if you see that we are recording this, I'll just give you a tail cuff method. This is used to. So initially when people used to think that, okay, we are recording the systolic and diastolic, because it goes peak goes like this, then there is a peak. So peak is exactly give a systolic pressure. People used to think that where there is a tapering and it is a diastolic pressure. It is not true. So we are only recording systolic. So similarly, what I said is when you put common carotid artery, you are going to record a mean arterial blood pressure. Okay. So here you can, in this experiment, you can record a pulse rate as well as, you record a, systolic pressure. Now we do not do it. We do a telemetry. So you happen to go to lab. So there is a telemetry where it records all the ECG, all the EMG, all the parameters it can be recorded. But here's some of the point I'd like to highlight, that when you record the ECG of the animal, when you compare to the human, we have a PQRS. So you compare the human heart rate, it is on an average is 72. So you can record this wavelength at this speed. But you take an example of a mice where heart rate is more than 450 or rat more than 300. So normally when you record it, you could find that ST segment is absent. So normally if you see there is a ST elevation, patient come to emergency, you get to know that there could be myocardial infarction. You call it heart attack. So only the difference. So you need to know that what is the difference between a human ECG or an animal ECG. So these are because of increased heart rate. Now we have a procedure called telemetry. As I said, you can record all the parameters. Otherwise, conventionally, you can just anesthetize and you put an electrolyte like lead 1, lead 2, lead 3, and you record the ECG. Now coming to one of these important aspects, as I in the beginning said, that we have many diseases where we do not have a medicine. So one of the diseases is autism. So since we do not know any pathology, there are a lot of hypotheses because of myocardial dysfunction or chronic neuroinflammation. There are a lot of hypotheses there. So here if you see that how do you create a model. So we use that we have several models like valproic acid model, propionic acid. You just read some of the story from Vietnam War. So after the Vietnam War, they have used some of the agent And after that, there is a prevalence of autism was very high because of this. Okay, because of water contamination. Similarly, you might have noticed that there are earlier a lot of Chinese toy used to come to the India. So recently there's a ban because that whenever you give any of the toys to the child, what is this? They hold it and they try to leak it. So it was found that the lead compound is very high. So these are a couple of hypotheses. So when you recruit the animal, so animal, the pregnancy duration is maybe 21 days to 22 days. So we just look at that in a pregnancy. When you talk to a doctor, you will tell you that do not use any medicine because maybe none of the medicine is very safe. So you say the stage of implantation, you say during the period of organogenesis. So during the period of organogenesis, we do not prefer to give any medicine except maybe iron supplementation because many of the people are anemic, or maybe vitamin supplementation. So here you inject 12.5 days valproic acid, then that pups are there after 22 days. So normally, as I say, that you screen the pup, you want to screen out the male pup because male are more prevalent. So you identify that how many of the pups that develop autism, like there's a swimming performance. If there is autism, then there is a delayed eye opening. There is a delayed eye opening. Then we have the olfactory discrimination. I'll tell you that whenever you give any food to autistic child, they'll not eat it, they'll smell it first. And if it is comfortable, then only they'll. So similarly, same information can be implemented in the male pups or female pups of the animal also. So you screen through the swimming performance. So if you try to walk and allow the pups to walk, it is not going to go in a straight line. It will go to the side and fall down. So these are some of this screening one. After 21 days, then you do a lot of battery of the test. So many of this problem in autistic is they do not speak, they do not communicate, they do not have any friend. So similarly, if you see that this is a chamber, 3-chamber, and they'll not go to another chamber to coordinate with any of the animal. Okay. So, but if you take a cotton and keep it in one chamber, they'll try to identify from smell that this is from the mother smell. Okay. So these are, you can indirectly say that this, particular pups is autistic, and you can accordingly, if you have a medicine, you can try it and you can evaluate it. So these are some of the experiments we do. And as I say, we have a vision that we screen the animal when you do learning and memory. And as I have already explained of 3 chambers, social. So there is a battery of tests is there for social behavior or behavioral tests. We have to do battery of tests. For social novelty tests, we can do it. Now coming to a very important topic is, how do you validate a model? So normally when you create a model, I have given an example of Alzheimer's disease. What do we do, Alzheimer's? We link to the pathophysiology. Is it beta amyloid? Is it tau protein? Is it diabetic link? So we have several models there. Now, when you select a model, you know that this is very important, that validation is how to validate. If you go back and see in 1969, it was started by burning. So unless you validate it, if you do a study, that it is not going to work it. So when you talk about validation, you need to need that you are comparing with a patient coming to the OPD, they explain when you take a history that I have this problem, then you examine. So you try to correlate that how much it will mimic the clinical situation. So when you talk about validation, we always talk about construct validity, phase validity, and predictive validity. So basically you want to see that how much clinically it will mimic. So this is one example that you say about predictive validity, phase validity, because you are taking a history in human and ultimately you're going to correlate. So these are the points are there: strain validity, early development validity, etiopathogenesis, and these all can be validated by the scoring system. Whether model is low, moderate, and high. And that is why you say model system validity, you take about the level of validity, organism complexity, and how much disease simulation is there. So you can take an example, just go through some of the paper. So you get to know that how do you score that this is a model, it is validated. How do you establish that? So these are the information that you can score it also. So this is what you do for it. You look for, just remember, how do you say how much is phase validity, how much is construct validity, phase validity, and predicted validity. So in quickly, you can say that you can score this, identify the model that how much it is validated. Please go to the arrive guideline also. So I'll not go in detail. And also you can say there is an internal validity or there is an external validity also when you read this paper. So then you can say that looking at the internal validity and external validity, how much it can be translational is there. So just quickly go through this paper. Now coming to the third important point is that once you are an experience and you want to go for a drug development process, how a regulatory authority look at the data. So as per our regulation, earlier it was Schedule Y, now it is New Drug Clinical Rule 19 March 2019 as a part of Drug and Cosmetic Act in 1940 and 1945. So when you say drug development process, go for a regulatory toxicity studies. So quickly, I'll just go through because it is almost going to be 1 hour. If you do a toxicological studies, you wanted to see that what I say that we wanted to see the biological variation. So if you give a drug, you wanted to see that identify is there any toxic hazard is there, and you wanted to quantify the assessment of risks to the population. So that is why that depending on the clinical use of the drug, suppose you give it for 10 days, maybe a good toxicity study is sufficient. But if you give a drug for more than 6 months, like in a chronic disease, like I take an example of diabetic mellitus, hypertension, Alzheimer's, where you have to give a long-term toxicity study. You have to generate. The reason behind, more you go beyond 6 months, the question is coming that can this drug causes mutagenicity? Can this drug causes carcinogenicity? Then that is why you have to do it. So there is a guideline. I think you just remember, you just read the story of thalidomide, where it was started in 1957 for pregnancy-induced emesis in vomiting. And it was found to be a very good drug. And it was introduced, and later on it was found that, there are 10,000 babies born with phacomelia. Then one of the pediatricians had written a letter to Lancet that I am a pediatrician. Normally I found maybe less than 1% congenital anomaly. But recently I have found huge number of seal phacomelia. And that's how this guideline has changed and the regulatory becomes stringent. That means you wanted to do a study in the regulatory toxicity that may not be one species sufficient, because when you talk about thalidomide, they have done it in mice, rat, but they had also done in rabbit, and rabbit they could find it is causes teratogenicity, but they hide the result. They're thinking that in a smaller animal it is fine. So when matter went to the court, they had to open up all the data. So we also see male and female. Is there any variation of drug response? If you look at the guideline today, earlier one, they say that we are going to do phase 1 only in healthy male volunteers. So you buy a medicine, you find a drug is, we do not have any information about female. We do not know how it is going to behave in children. So because it has never been studied. In 1994, Canadian government has come up that we should also go for a female in the healthy volunteers, provided they have adequate protection for pregnancy. So that's how it changed the guideline that we need to know the species variation. We need to know that how the drug is behave in male and female. So that is why here in animal toxicity studies also we include male and female. We do a lot of PKPD studies also, and number of sufficient animal, and all the SOPs is maintained. And most important thing is all these studies need to be done in the GLP facilities. Where is it, the GLP facility? So this is what a good toxicity study that if you take an example of paracetamol is a commonest poison for liver toxicity, but still we use because we exactly know that how much dose is going to cause poison. So similarly, when you use intermittently, we have to generate the data for subacute and subchronic also, and long-term toxicity study. So this is a process for drug development. I'll not go in detail. And however, when you do complete that preclinical toxicity study, you get to know that you have to make an application as an IND, investigational new drug. That is for that you completed a study of regulatory toxicity study in animal. Now you're planning for a first time in human. So in India, we have that any drug which is discovered, it is allowed to do phase 1 study. But if you discover a drug outside India, they have to go for the initial phase outside, then they can come back and repeat the test. So IND is not an application for marketing approval. So these are the tests are there: acute toxicity, repeated dose, safety pharmacology, pharmacodynamic, ADME. Basically, you wanted to see that how the drug is absorbed, how the drug is distributed, how it is metabolized, how it is excreted, genotoxicity, reproductive, all special toxicity studies, and even the drug you are giving, can it be phototoxic? Can it cause irritation to the eyes? Or in children, we use some of the rectal preparation, so we have to generate the data. So quickly, if I say acute toxicity study, we wanted to see that, all these clinical signs, and we observe, give the dose, observe the animal for 14 days. So this is how we do. So what do we extract from acute toxicity study? Basically, from this acute toxicity study, we extract what dose could be go for a human dose, like NOEL, no observed effect at that level, or we extract the LOEL. So low or adverse effect at that particular. So how it is going to helpful is, as I already said, that we wanted to see the species variation. Also, that if the drug is toxic, basically we wanted to see that when we sacrifice the animal, there are 47 organs, but we are more concerned about vital organ. Is it affecting brain? Is it affecting lung? Or is it affecting liver, heart, spleen? So like that, all the vital organs we see, and we go for a morphology and a histomorphology. So that is what we go for a NOEL and LOEL from acute toxicity. Now, you quickly take an example of you work on ABCD. When you plot the toxicity data, you might be thinking that C is better than the D because it looks like more toxicity. But just go deeply, C is more toxic than D, and that is why comparatively when you work on several compounds, then you get to know that no, C is more toxic than D. So that's how we have to interpret a conventional toxicity study. Then we go for repeated dose toxicity study. You go for 28 days, 90 days, 6 months, 1 year. Here also Beckel is used. So ultimately, if you see that it talk about how it is going to have different organ like skin, mucous membrane, CNS. So you go for hematology, body weight, histopathology. So what you wanted to see that if there is an ADR also, because any of the medicine, it has to be ADR. You cannot say that zero ADR. So that is what we also, confirm the level of NOEL. Then you have a genotoxicity studies, in vitro study, in vivo study. Then carcinogenicity. Carcinogenicity, as I said, that if you use any drug for more than 6 months, or earlier Me2 drug shows some problem with the mutation or carcinogenicity, then you have to go for a long-term toxicity study in a chronic situation where in clinically you have to go for more than 6 months study. So these are the tests is there. Only difference in acute toxicity or repeated dose toxicity, in carcinogenicity, you have a positive control there. And you also go for a biomarker. Like you say, biomarker for alpha-fetoprotein or HCG. So these are studies are done. Then we have a safety pharmacology study, huge number. Reason behind, if you take an example, that right now we do not have terfenadine, because terfenadine has been withdrawn because of QT prolongation, heart arrhythmia, ventricular arrhythmia. And that is why it has been recommended new guideline. Any drug you develop, you have to see that can it cause QT prolongation. So we have battery of tests. So here, along with this safety pharmacology, you also see, can it affect the respiratory system? Can it affect, suppose you plan a drug today and you go for a phase 1 study. So you get to know that the healthy volunteer come back after 1 year, say that I was not married, but now I got married and I'm infertile. So he might refer it to it is because of drugs. So and that is why we have a lot of reproductive toxicity study, whether it is affected immunity. So all these studies have been done. So there's a regulation as per Schedule Y earlier. Now it is clinical trial rules. So accordingly, you have a number of animal. If we use for clinical situation 10 days, you have to generate the data in animal toxicity 3 times during the animal experimentation. So these are special toxicity study. How do you say this drug is safe in pregnancy? So you have a segment 1 study, segment 2 study, segment 3 study. So as I say, can it affect the implantation? Can it affect the during organogenesis? Or when there is a lactation, is it going to affect? We have a perinatal study. So these studies are there, male fertility and female fertility. I'll not go in detail. And also, because once you give a drug, then you go for, PUPs. So these studies need to be carried out for teratogenicity or perinatal study. As I say, segment 1, segment 2, segment 3 studies. So these are done. And also dermal toxicity studies, especially you can use rabbit. And also guinea pig is used because guinea pig is more sensitive to histamine. So that is why vaginal toxicity study, rectal tolerance studies. So all this. In case of a parenteral one, like you take an example of vaccine. Okay, you are giving, intramuscular. Is it going to affect the site of, injection? So these studies are already done. Then we have any of the ocular preparation, especially we use rabbit, and we can also do ocular kinetics. So if you see that if you give a drug and there is a redness of the eyes, you get to know that redness, if it is there, that means it is irritant to the eyes. So vessels are dilated, you can get to know it is red. So these are indirect evidence for, and similarly for inhalation toxicity studies also special preparation are there. Allergogenicity, hypersensitivity I have already talked about. Then we do studies for lymph node assay also. Like you may take an example of phenytoin. Phenytoin causes pseudo lymphoma. So initially, when you do an experiment in the drug development process, you get to know that it is going to affect the lymph nodes also. So then accordingly, dynamic studies, because as I say, we do PK, PD study. There is several models are there. Then there's a PK, PD also, pharmacokinetic, pharmacodynamic, or everything is correlated with the software. So here it is important point, QT prolongation, none of the drug is clear if it is affecting the cardiac arrhythmia, because there are several examples of withdrawing the molecule from the market, like I've given an example of terfenadine, and that is why. So there is a guideline for this, how do you go for a regulatory toxicity study to make an application for IND also. So these are for US 1571. India, we have Form 44 earlier. Now it is everything is digital with the new government, and also you can submit to the Sugam portal. So these are recommendations from the earlier guideline. Now it is still existing. How many of this animal need to go for it? How many number of animal it is required to go for a regulatory toxicity study? So when you submit this application, ultimately you know that you make an IND application. It is reviewed by medical team, chemistry, pharmacology, toxicology. There is a statistician is there. So initially you go for a safety review. Basically, if you say that you do a studies, you find there is an elevated liver enzyme. So what your next question? Is it reversible, irreversible, irreversible? If it is irreversible damage, you are not going to allow the drug to go to the next phase. But if it is reversible, I take an example of paracetamol. You take 1 table of paracetamol, 2.5 times more liver enzyme. But still we use because it is reversible. So accordingly, we evaluated. And in India, we have an IND set where it has been discussed. Then you give an approval for phase 1 study, first time in human. Once they complete phase 1, phase 2 in patient and phase 3 in a larger patient, you give an approval for NDA, new drug application. Then the drug come to the market, and then there is a surveillance program start. We have a pharmacovigilance program, but there is a PSUR, periodic safety update. Every 6 monthly, they collect the information, submit to regulatory authority, because we call new drug till 0 day where it is marketed till 4 years. So that's how it goes, cycle is goes. So as long as the medicine is there, we have to have a surveillance program for all the medicine. With this, I'd like to thank you. I think I'm on time. And if there is any question, I'll be happy to answer. Can you hear me?
Thank you so much, sir. Yes, sir, you are audible. From the participant side, there is a one question, sir. Dr. Priyanka ma'am has asked that as per acute dermal irritation study in rabbits, what should be the endpoint? Should we do rehabilitation or euthanasia, because OECD guidelines are
No, that is dependent. If you want to go for a histopathology, you have to sacrifice the animal. Because you go for a evaluation, suppose there is an ointment you apply, and you do the studies, then you sacrifice the animal and do the histopathology. Normally it is done. Then you are going to sacrifice under euthanasia. I am saying in case of a pharmacokinetic study where you need not have to go for sacrifice, you only take a blood sample for rabbit, so then you rehabilitate. Any other questions?
Thank you, sir.
Thank you. May I leave?
Any other question from the participant side? Thank you so much, sir, for sharing your, for gracing your important work and sharing with us your knowledge and findings today on the validation on preclinical testing, CPC guidelines, phases of clinical trials, and very well pictorially explanation of the animal handling. So thank you so much, sir. We are blessed with
Thank you.
to have you. Thank you, sir.
Thank you, all the participants. Thank you. I am just leaving now.
Okay, thank you so much, sir.
Thank you.
Um,
Ma'am, is there any other questions from the participant side? Or Vishwaji, can we unmute the participants so that the participants can directly communicate with the sir?
uh, Sir, one has asked the specific period for toxicity. The name is not mentioned, HP is written. Sir, I think sir has, Vikas sir, are you there, sir? Okay, so we'll answer you afterwards after having some emails to this from the sir side. Okay, so now we will be moving for the next speaker, Dr. Deepti Kakkar ma'am. So she will be joining us in shortly, so we have to wait for her.
Deepti ma'am is going to be joined in the 5 minutes. So we have to wait.
Okay, okay, so we will be waiting for her for 5 minutes.
Yes.
Thank you for your patience, participants.
Sorry, I couldn't hear it. Any questions is there, Dr. Nitin?
Anjana madam, there was a question. That time Vikas sir mic was muted, so you can repeat.
बता दो, I'll just answer it. Dr. Nitin, any questions?
So there was a question.
Yeah, can you repeat the questions?
Participants want to ask the period for the skin toxicity.
Okay, actually, that is as per the protocol. Na? So you wanted to, depending on the drug, so you apply it, then how long exposure it is required. So you look at the kinetic profile, that is it going to, absorb to the systemic circulation, everything you look at. So you give exposure for 24 hours, 48 hours, depending on a compound. Suppose in clinical situation, you want to use for a longer time. So normally we say that, can it affect the skin in a single application or when you continuously use it? That way you plan the, like suppose chronic urticaria, you use for a longer time. So otherwise, simple, you use 1 to 3 days. That's how you plan it.
Right. So Anjana madam, is there any more questions? Or can we conclude the session? Okay. So thank you, Vikas sir, for joining us and delivering this such a wonderful session in the 5Ds FDP programs. And I'm
Thank you.
expecting.
And next time I request you to include a GLP lecture also. That will be fine.
Sure, sir. So I will be reconnect to you whenever this FDP will be completed, and we will have a special lecture on the GLP, because we are in the process of making our animal facility and want to convert the facility into the GLP. So definitely with this objective, we require your blessing and your guidance.
If you have time, last day I can take for half an hour. That also can be done.
Sure. So for that, I will reconnect to you. And thank you very much for joining us this FDP program for this with this short notice of period. Thank you once again, sir.
Thank you.
So I would like to request all the participants, please join us, because our next speaker is going to be joined very soon.